Okay, 11th grade, here we are, lesson number 4 of our unit on experimental design, and we're going to be talking about precision in experimental design today, and we're talking about how to really narrow down your topic, your idea, your question into a really, really solid research question and look at the different kinds of hypotheses, your null and experimental hypothesis as well. This is the last lesson before you choose your topic and kind of lock in your question. From the so we started at 10, down to 5, down to 3. For today, this week, you need to be finalizing your topic and getting started to collect data, which is really exciting. So, first thing, as you've been narrowing down your ideas, you should be starting to kind of draft your questions. It's one of the tasks I asked you guys to do for each of your topic ideas was to draft your questions. And a lot of the time on the IA, students will lose points because their research question is not focused enough. So what I have here is screenshots from the rubric from the IBO about the research question. So it's at a 6 points, part of the exploration grade you get, it's at a 6 points. So if you get 1 or 2 points, if you say the topic of the investigation is identified and the research question, if some relevance is stated but is not focused. So if your research question is way too general, that's gonna knock your whole exploration score down to a 1 or a 2. So remember in the IB rubrics, your lowest kind of level you achieve, 1, 2, 3, 4, 5, 6, that's the level you get for everything. Even if you did a really good exploration and every other component of this rubric was at a 5, 6, if you got a 1, 2 on your question, that would drag everything down. So let's not let that happen. So your question should include both your independent variable, dependent variable, and the relationship between those two variables that you were investigating. So what are you looking at to measure to see the relationship between them? If we look at a rubric here, to get a 3, 4, the topic of the investigation is identified and a relevant but not fully focused research question is described. So this is not at all focused, this is not fully focused. And we'll look at examples of what this looks like later on in today's lesson. And to get a 5, 6, the topic of investigation is identified and a relevant and fully focused research question is clearly described. So the research question is very important for your grade. So a common format to use for making this kind of question, or a good way to start, is how does blank affect blank? How does the independent variable affect the dependent variable? This is a little bit of a simple place for your final question for DP students. I use this template a lot with my 9th and 10th grade students, but it can be a really good place to start to help you make sure you have the most important critical components there, and then you can elaborate on it, make it a little more detailed, even more specific, more comprehensive as you go. Again, we'll look at several examples of this today. Okay, so one way you should be specific in your research question is with scientific names, with the species names. So in topic 5, we talked about Linnaean classification system. We went from domain, kingdom, phylum, class, order, family, genus, species, all the way down. And we talked about that already. Hopefully you remember some of that. It's coming back to you. If not, take a minute and look at your notes. So part of having a really specific and focused research question is having the correct scientific name for any organism you're going to choose to work with. This most often happens with students working with plant species that they have to choose because, of course, according to IBO ethics, we can't work with animals in any way that would harm or stress them. So it really only leaves with observational studies, which aren't done very common. I think some of you are looking at doing observational studies for your IA, which is great. So you need to know the species name of the species you're going to be observing. And this is really important because common names, they can have many, many common names for a single species. Okay, so here we have a pebble crab is one common name for this crab, but it only has one scientific name, no matter which language you speak. The scientific name is Xanthias lamarcki, and that is, if you are a French speaker, if you speak Arabic, if you speak German, if you speak Mandarin, whatever language you speak, that is the scientific name for that species. And it's the same for everyone everywhere, and that's really important for communicating and being clear. So if you're choosing to use some sort of organism, whether it's a plant or an animal you're going to be observing, you need to make sure you use a scientific name in your question and throughout. Now, when you're writing this name, you need to make sure you have the genus name and then the species name. Okay, so here's some examples. Panthera pardus and Felis silvestris are two different cats in the class Felidae. And you notice the format. When you type a scientific name, it's always italicized. So italicized it here, it's always italicized. If you write it by hand, which you won't really do for your IA, but it's a good habit to get into for IB exams and unit tests and stuff, you underline the name. You always capitalize the genus name. So here, look, Panthera is capitalized, Felis is capitalized, Xanthias is capitalized, and you leave the species name as lowercase. So Lamarckii is lowercase, Sylvestris lowercase, Pardus lowercase. So make sure you make a note of how to do this, and if you choose to use any sort of organism in your mock IA or your real IA you do next year, you have to use its correct binomial nomenclature scientific name. Okay, now in every single investigation, let's go back to variables again. You need to have one and only one independent variable. Why is that the case? Well, if we want to know what's causing something and see if we can have some sort of clear relationship between the variables, we can't manipulate tons of things. So in both these examples of plant experiments, they have changed the number of drops of fertilizer, but notice the water amount is the same, the light is the same, which is the number of drops of fertilizer that is different. We have a control group with no fertilizer, and then we have 2 drops, 4 drops, 6 drops, the experimental group. Here we have different kinds of water, so salt water, distilled water, and tap water, different kinds of plants. And they notice the controls, the constants, the same size container, same potting soil, same room temperature, same temperature of water. And we're looking at the height. So if we manipulated more than one thing, let's say in this plant experiment, we manipulated how much light and how much fertilizer, how would we know which one was causing an effect in that plant? We wouldn't know. So we'd never do more than one independent variable. Now that being said, it can be useful to include more than one dependent variable. That means you're measuring more than one thing to see what the effect the independent variable has on it. Now we don't normally do this with high school investigations because it's a little more complicated and we want to focus more on kind of the foundational elements of experimental design. But you guys are DP students. That means you're doing something a little more intense, a little more complex than you would be with if you weren't in the DP. And so you might be looking at more than one dependent variable. This is really handy for biology specifically because if you're working with an organism, it's really good to have multiple dependent variables. Why is it about organisms is it good to have more than one dependent variable? Like as we said a bunch of times, it's really hard to predict and control everything that affects an organism. So for example, in these plant studies, if my dependent variable was height and only height, and that's all I could get data on, what happens if there's no real pattern in height? What if this particular plant species, or this one, is not affected by the fertilizer doesn't affect height, the fertilizer focuses more on root growth, or lateral growth, side-to-side growth instead of vertical growth. So if you have more than one dependent variable, then let's say you're collecting data on a plant on height, on biomass, on the stem width, on the number of leaves, on leaf surface area, a variety of things. You can choose, usually no more than 3 is kind of recommended number. Then you have kind of backups. So if what you're expecting to happen doesn't affect the dependent variable you've chosen, you have other variables you can look at. Now, once you complete the investigation, you have all that dependent variable data, you have a couple choices to make. You can choose to look at those and say, okay, well, I didn't see any change or any difference in height, but there's definitely a difference in roots. So I'm going to change my question, and I'm going to focus more on the root component of growth. Or you can analyze multiple and say, look at this, there was no relationship with my statistical test. There was no relationship between fertilizers and height, but there was one between fertilizers and root, or fertilizers and biomass. You could also do a investigation to see how multiple things affect each other, and that's called an ANOVA test, and we'll look at that later on in this unit. Because of course, if a plant has bigger roots, that's probably going to make it grow bigger in general because it has more access to water. It's going to be more stable kind of foundation for the plant in the soil. It can grow taller, all those kinds of things. So a lot of these things can be interrelated. So this is why often we don't allow students in high school to do more than one dependent variable, but it's for the DP is totally fine. And if you're thinking of working with an organism, I really recommend you pick more than one dependent variable and track it through your entire investigation, and then you can choose what to work with. A lot of scientists and graduate students always have more than one dependent variable, especially if they're going to be investigating anything with humans, like psychology uses a ton of dependent variables because it's really hard to predict what's going to affect what. And you can look up some really cool examples of studies about this. Okay, let's talk about hypotheses. We talked about it kind of in general before. Let's get a little more specific here. So here's a theoretical investigation, and we'll look at data for this investigation later, about goldfish, how goldfish population density, so how closely confined the goldfish are, how that affects the length of the fish, or the average length, mean length of the fish. So what could the research question be? Well, here's an example. How does population density affect mean fish length in Carassius or Aratus? So I have my independent variable as population density, my dependent variable is fish length, and I specified the species of goldfish. That's an okay question. It could be better, but it's a pretty good question to start with. And then I'm going to have my hypotheses. So when we're doing statistical analysis, we have to have a null hypothesis. We looked at this a little bit with chi-square test earlier in the year during our very first unit and the quadrat sampling lab we did. We're going to come back to it with our statistics in this unit as well. So what we normally think of as a hypothesis is called the experimental hypothesis, or H1. And this is where you're going to predict the answer to your question. And so if this is the question, what would your hypothesis be? As population density increases, mean Carassius Aratus length will decrease due to the lack of space for growth. So notice how I'm explicitly answering the question, what I think will happen, the length will decrease, and I have a reason why, due to the lack of space for growth. It isn't super long. I've seen some students write hypotheses that are 3 or 4 sentences long. That's not a good practice. That kind of information should be in your background information, your introduction, not in your hypothesis. So that's your experimental hypothesis. Now, when we're doing stats, we have to add the null hypothesis. So the null hypothesis, what do you think the word null means? Well, null means not, or non-existent, right? So the null hypothesis, or H naught, H a little zero, states that there is no statistically significant difference between the samples. Notice it's not saying there's no difference, there's no significant difference. You have to do a statistical test and see if it's significant or not, the difference between them. It doesn't mean samples are identical, but it means the difference that there is due to variation alone and chance alone, and not due to some relationship between the variables. So for the fish experiment, what might be the null hypothesis? There is no difference in mean length between the denser and less dense populations. Okay, there is no difference. That's the key component of a null hypothesis. Okay, so let's practice a little bit here. So here is a really useful checklist of 8 things to make sure your question is focused, especially for the IB, for the IA. And there's 8 kind of steps to follow through. So we're going to do a practice one together going through all 8 steps. And the question is, does changing the pH levels have a measurable effect on photosynthesis in plants? So we're going to work through all these 8 steps to make this question, it's very unfocused, to get 1 or 2 points only on the IA rubric and make it much better. So we're going to first start with deciding the idea or topic of study, looking for variables and their ranges, decide on the DV and the IV, check the IV actually causes a change to the DV through biology, so you have to do some research. Is that a relationship that actually exists? Something might happen. If the DV is a calculated value, identify what will be measured. Squeeze the scope, focus the question on specific things, so narrow it down, like what species you're going to be working with, that kind of thing, what pH range. Make it scientific, correct units, scientific names of species, biological terms, that kind of thing. And see if you can describe a null and alternative hypothesis. Alternative hypothesis is another name for experimental hypothesis, but it's the same thing. Okay, so here's an unfocused question. Does changing the pH levels have a measurable effect on photosynthesis in plants? Well, the topic is clearly photosynthesis and how it affects, how it's affected by pH. The variables are pH, is my independent variable, And the pH range, of course, is from 1 to 14. And then my dependent variable is the rate of photosynthesis. The dependent variable, rate of photosynthesis, because the rate varies depending on the pH, but not vice versa. So the pH does not depend on the rate, but the rate does depend on the pH. Okay? How can pH cause a change in photosynthesis? So we're checking the science. Is there actually a relationship, a theoretical relationship between these two variables? Yes, we know that enzymes are involved in photosynthesis and that pH can change the rate of activity of enzymes. We know that to be a fact from our lessons of photosynthesis, which is our last unit actually. Okay, the fifth step is a derived value. So photosynthesis rate is a derived value, so it has to be numerical somehow, and we calculate it from something measured in the experiment. So your choices could be bubbles of oxygen produced, the rate of change in carbon dioxide concentration, or you could take 1 divided by the time taken for a spinach leaf disc to rise. So there's an experiment for photosynthesis where you have little discs of leaves and they rise as they lose matter, as they lose lose O2, and makes them kind of float. So that could also be one you could do. But you need to pick how you're going to actually measure this. It's something that's measurable, like photosynthesis. How will you measure it? Okay, step 6, squeeze the scope. Name the species of plant. So example here, spinach. They chose spinach for this one, and here's a scientific name as well. And squeeze the scope, so narrow it down by specifying the range of pH values from pH 1 to pH 7. So they're doing all the way from 1 to 14. We're just doing the acidic part of the pH scale. Okay, so here's kind of another draft of that question. Does changing the pH from pH 1 to pH 7 have a measurable effect on the rate of photosynthesis in spinach plants? Spinacia oleracea, estimated from the time taken for leaf disc to float. Now that's not the only possible question, that's a very good question. And then double check that you can go with a null and experimental or null and alternative hypothesis for this question. So a change in the pH will not change the rate of photosynthesis, that's your null. And as the pH decreases, the rate of photosynthesis will decrease as well, that's your experimental. Okay, so having gone through all those 8 steps, what's the new question you would write for this particular? So this was our original question. How would you rewrite it to be much more focused and get us out of the 1-2 point range on the rubric and up to the 5-6 range? Well, here's my example. How does changing the pH, range 1 to 7, affect the rate of photosynthesis in spinach? Oh, not in the spinach, just in spinach. Look at this, full of typos. Spinacia oleracea measured by oxygen production. So instead of doing the spinach disc to float, I did oxygen production with an oxygen probe. You can do that as well. But notice I clearly have my variables, I clearly have how I'm going to measure things, I have a species, it's much more focused than it started off being. Okay, let's do some more practice here. So here's a list of 5 questions. Some are more focused than others, and I want you to arrange them in order from the most focused to the least focused question. So pause the video, take some time, and put them in order from the most to least focused. So the most focused one definitely is D, because in D we have, in D, we have a duckweed that's a specific species. It's a common name, not the scientific name, but it's an actual name of an organism. And the length of time, 12 days. We have pH, how does it affect the growth of duckweed. This can be a lot more specific, but it's a pretty good start. Then the next one, in my opinion, is C. So I have specific peppers affecting pulse rate. But saying their ingestion, I'm not saying like how I'm ingesting them or how long after pulse would be affected or any of this. It's kind of vague still. The next one is B, and B and C are almost similar kind of levels of focus. What effect does changing the amount of sugar in a yeast solution have on the respiration in the yeast cells? So how will you measure respiration? Are you measuring with CO2 production? Are you measuring it with O2 being taken in? How are you measuring, like what range of sugar are you putting in? What species of yeast you're going to use? All these kinds of things. and what concentration of solution you're going to use. So this is less focused because it has pulse rate already tells you how it's going to be measured. Right? You're measuring pulse rate only one way by counting the beats per minute. Whereas respiration is lots of ways you can measure that. So that's why this is more focused than this. Next one is E. Is there a correlation between the ability to do tongue rolling and the ability to recite tongue twisters? So tongue rolling is a genetic trait. Looking, is there a correlation? But it's not saying like, how does one affect the other? They're not saying how they're going to measure it, any of these kinds of things. So it's not super focused. And then A, this is the least focused one. How does gender affect how well one can recognize assent? It says very unfocused, and not a particularly accurate and testable question because it's really hard to make sure there's no other factors affecting this except gender, and how you're using your particular idea of what assent smells like to cause someone to identify it. So it's a tricky one to choose to do. Okay, so I want you to write out for the 8 steps above how to do this question. How does gender affect how well one can recognize assent? That's the worst one from the previous slide. You need to work out for me all 8 steps in your notebook and on your typed notes. Identify number 1 is this, number 2 is this, for each one like we did with the pH and photosynthesis question, and then write a better question for it. So the steps that make it scientific, I want to see that part of it. I'm going to give you feedback on that. So taking a general question and making it specific for and appropriate for an IA. Then you're going to take these 4 other questions, and for these, you don't have to do all the 8 steps. All you have to do is make it better. So take all these other 4 questions we already looked at that we said were not very focused, and make them focused. So you can use the 8 steps to remind you of all the things you have to include, but you need to write them out for me step by step. Just write, this is my original question, here's the new one. Original question, here's the new one, and I'll give you feedback on how that goes in the I'll go to Schoology. Okay, so the next step, far from the last step, the next step for your IA is choosing your mock IA topic. So I'm not going to ask you to just choose it and post it in Schoology. I'm going to ask you to start an email conversation with me. I know we get a ton of emails. I feel like hundreds every day. You guys get a lot too, I'm sure. But I want to have an email chain so we can discuss this back and forth a little bit easier. So I'm asking you each to email me what your choice is. If you're debating between 2 or 3 still, then send me those 2 or 3. We can talk it out together back and forth. Make sure you consider what you can do in quarantine. What can you get? If there's something you need to order, maybe it's some plants, can you get it somehow? I know there's a vendor that's still selling plants in Agdal up a little, the municipal market, and you can buy plants there. Can you order stuff online? I just discovered Jamaica. Can you order stuff there if you need? Do you have it at the house? Do you have what you need, or can you get what you need to do the investigation? What did you find good sources for in our last class about research? And what can you really have a really solidly well-designed investigation around? Remember, I'm happy to help you, and I can help you. I can't tell you what to do, but I can answer your questions and kind of guide you and give you feedback. And remember, this is your sub. There's no unit test this unit, so this is a really big part of your grade for this unit and this year, because RAS's assessment policy is to use the most recent and most consistent grade. So if you're someone who's been getting like 4, 5, 6 is kind of around that range, or you're getting 3, 4, 5s, if you can get like a 5 or a 6 on this mock IA, that will really increase your overall grade at the end of the year. Okay, so do the best you can possibly do, and that starts with a good choice. Okay, so your homework is upload your notes so I can see your thoughts on this problem. Remember, do this half-screen notes like you're always supposed to be doing in this unit. It should include the rewritten questions. So the first one that we did, the gender and scent, write out all 8 steps for me. The other 4, you can just do a new and a better question to write the steps. If you want to, go for it, but you don't have to. Okay, and that's the end of lesson number 4 for this unit. I hope you guys are enjoying it and you got lots of good ideas for your mock IAs. Take care.